start with the phase of cell cycle. So these consist of two phases basically: interphase plus mitotic phase, which we can say M phase also. In interphase, the cell cycle takes around 90% of the time. Interphase, we can say interphase is the building time or building phase of cell. In mitotic phase, it is a phase of cell division. The cell divides here. Okay, so basically in interphase, it is divided into three phases. For example, G1 phase, S phase, G2 phase. G1 means gap 1 or first gap phase. S means synthesis phase because DNA forms here, chromosomes build up there. G2 is gap 2 or second gap. Okay, now we talk about the mitosis phase, mitotic phase. In this one, the cell divides here. It is the shortest phase. For example, like if this phase takes 90% of the time, the mitotic phase, or we can say M phase, takes 10% of the time for the cell division. Okay, now the question arises, why G1, G2 are called gaps? They were called gap because like at first when they was observed in the cell, there were no activity undergoing in them. But today we know about that we know that the metabolic activities and cell growth are done here today. So that's why at earlier they were called gaps, but now they are not. So what happened next? Okay, in interphase, what does 3C denote? In the G1 phase, known as gap 1, the cell growth occurs. Cell continues to grow here. Okay, then the S phase comes, synthesis phase. Here what happens? The DNA get divided into chromosomes, get divided and transformed into chromosomes, but we cannot see them because they are not coiled here. Okay, the next come is G2 phase, the second gap. In this phase, like the cell grows further and final preparation are done for the cell division. Last, what is left? M, the last, which is known as M phase. Here the division occurs, the cell divides properly. So again recall the steps. First comes the interphase, then comes the gap 1. What happened here? Cell grows, then the S phase, DNA doubles and replicates, and it converts into a chromosome, but they are not condensed yet. Okay, but they are duplicated. Okay, then second is gap 2. In this means final preparations are done for the cell division. Then the mitosis phase, where cytokinesis and mitosis take place. Okay, then what happened then the two daughter cells formed here, and one of the cell like continue to divide again. So like it forms a new cycle again and again. and the growth and the life continues. So our next question arises: how much time does this all cycle take? It takes 24 hours to divide. Which is the longest phase I told you earlier it is interphase. It takes 90% of time. How 90? Let me show you. The M phase is the shortest one. It takes 1 hour only. In the S phase, it is the longest one. Why? Why it is the longest? 10 to 12 hours. Why? Because the DNA is converting himself and the chromatin is converting to chromosomes. This take time, but they are not. We cannot see it right now because they are not condensed. They are uncondensed form. At here, DNA is uncondensed form. We can say it like this. It is not coiled up. Then comes the G2 phase, final preparation for needed for the cell division. It takes 4 to 6 hours. And the last one is G1, the first phase. It is like variable. It is sort of variable phase. Why? I'm saying that because in some cells it take 4 to 6 hours, or in some cells it take 5 to 6 hours. This is because due to like some cells in the multicellular organism divide very infrequently, or sometimes they don't divide at all. So that's why here it can change, like it can change to 4 to 5 hours or 6 to 7 hours. So it's different. Sometimes what happens is the new topic coming, like the cell undergoes a phase which is known as G naught phase. Like here, the cell does nothing, the division stops. It's not dividing here. Why? These cells, like for example, in G naught phase, these cell like doing some task, for example, uh, secretion of juice in gallbladder. Started to do these tasks instead of dividing and growing. Okay, so that's why in sometimes a gap occurs here and the cell cycle stops for a few hours. So that's why it is like it is the most like so that's why it is the most variable phase. Now we jump into the next section we call as mitosis. What happens in the cell division? So there are 5 phases for that. First one is prophase, second one is prometaphase, third one is metaphase, fourth one is anaphase, and the last one is telophase. This happens in mitotic spindle. We'll learn about it today. For to remember it, you can use an abbreviation known as P2MAT. P for prophase. P2 here means prophase, then prometaphase. Okay, then metaphase, anaphase, telophase. So let's make a cell for better understanding. For example, this is a cell. Okay, this is cell membrane, or we can say it is plasma membrane. Wait, plasma membrane, SPM. Okay, this is nucleus. Okay, and inside of it you can say it is nucleolus. Okay, and this protective covering of nucleus we know call it as nucleus envelope. Okay, and there are few more structures. For example, this one. Wait. Yeah, these are known as centrosomes. Okay, it has each centriole pair. This one is known as centriole. Okay, triole pair. There are two centrioles, so it means a pair. Now inside of the nucleus, there are chromosomes. Okay, these chromosomes are duplicated, but they are not condensed. Not condensed. Okay, so like what basically happened here, after the S phase completed, in the interphase when the S phase was completed, this is the result. This is G2 phase, final phase before mitosis. Now what will happen here, these centrosomes, sorry, have two pairs centrioles. They form microtubules, and furthermore it take part in cell division. From here it is a crucial step. Chromosomes, or the replication of DNA, took place in S phase, but they are uncondensed form. They are not coiled up right now, so that's why we cannot see it under the microscope. They are not visible to the human eye right now. For deep explanation of this phase, I have a better diagram for this. You can see the interface here. This part of interface, and the chromosomes are getting duplicated. Okay, and the centrosomes are making a form of esters. I'll explain it to you. And this is the final phase. After that, what will happen? These form the array, then they form the microtubules, and a cell divides in half, two daughter cells. It's a long process, but yeah. To begin with the first phase of cell cycle for cell division, we termed it as prophase. Okay, so what basically happened here, so you can see the chromatin started to getting condensed. Okay, the chromatin fibers are getting condensed. You can see there chromatin fibers started to getting condensed. Furthermore, what happened is the centrioles have started to flow the kinetochores, or the microtubules. They're starting to flow the microtubules towards each other and forming a early mitotic spindle. You can say it, early mitotic spindle here. Now in this case, we can observe this chromosome under the microscope, because like they're starting to condense. They're starting to condensing. You can see here the main event also occurred, like the nucleoli, the nucleolus. It is disappeared. Let's write it. The nucleoli or the nucleolus disappeared. Now in this one, like each duplicated chromosomes have the centromere attached here. Okay, they are joined at a point. It's called a centromere. Okay, now we can see the two sister chromatids here right now. For example, first sister chromatid, second sister chromatid. We can observe in this one. As I told you earlier, some of the arms are joined by the cohesion. We know called it as sister chromatid from cohesion. Another thing I like to mention is about esters. So what are ester? Ester are the thing when the microtubules from the centromeres, the centrosomes, are directed toward each other. This is termed as ester. I read the definition for you. The areas of the microtubule extend from the centrioles and like they are forwarding toward each other. You can also see like the centrosomes are appear to move forward in opposite direction from each other. So this resulted in the mitotic spindle. For example, this one. In this one, you can see they are closely packed right now. They are side by side, but in this case, they're getting far away from each one. So yeah, we can say formation of the mitotic spindle has begun.